biology teacher's favorite sample to introduce people to a microscope on how do you make a slice or a slide that it has a very thin slice of your sample is an onion. So you might go, oh, okay. So here's an example. Suppose that this saran wrap is an onion peel. So when you peel off the onion, if you do a good job, which I'll show you how that is normally done, then you end up with something like this. When you look at the microscope, you would see like each individual cell, and you could see like the nucleus. If it's a yellow onion, then you'd have to put some stain on it, so that gives it some color. You might still be able to see, if you play with the light, you can see the contrast of the ends of the cell, but it's easier when you put a dye on it. If it's red onion, then it's already pigmented, and you don't actually have to add any dye. So this thing here, the saran wrap with the cells that I drew, they represent a good slide that was made with the onion skin. Now, what happens when the slice of onion that you took was too thick? Or you could even think, I did a good job making this slide, but the little thin membrane of the onion folded over. So if the membrane folds on itself, then it would look more like this. So this part here is a good part of the slide because you can see individual cells. This part here is where the onion folded over. And then it looks kind of messy. You're like, oh, I cannot really make out which cell, where's what. And then you could make it even worse if you made a thicker preparation. So you could look at this and you're like, my gosh, what's that madness? And in the exam, I'm going to give you pictures of cells, of onion cells. And then I'm going to ask you, what does the vacuole look like? You're going to learn about it in lecture about the cell structure, but the vacuole is inside of the plant cell holding a bunch of water. And if it loses water to the environment, it shrinks. So the question in the exam will be, is the vacuole full, or is it shrunk, or can you not tell? So if you get a part of your slide that has single cell layer, you'll be able to see the vacuole. If you get something like this, then it's like, are you crazy, Dr. T? I have no idea. I cannot see the vacuole. It's madness. And that's when I'm like, okay, good. So tell me you're unable to tell. Not because of your lack of ability, but because the slide is not very good. So I ask you, why can you not tell what is the status of this vacuole? And you're supposed to say, too many cell layers, right? Like the sample is too thick. Some people say it's not in focus, but actually if you see sharp lines, then it is in focus. The problem is too many lines. All right, got it? Are you gonna get that question right? You could be. I promise to you it's gonna be there. Because thick samples are often a problem for a microscope. So when people want to make samples that are not onion, sometimes you have to embed whatever it is that you're gonna look at in some resin, and then use a microtome to thin slice it. All right, good? Now you know the importance of thin preparations, and now you know why.